My name is Astha Symes, and welcome to Live Longer World. I know, Min, you started off with your work in trauma surgery and as a physician. So I'm curious, maybe if you can touch a bit on your background and how you got interested in bioelectricity.
Yeah, this is a kind of a long story. So I was trained as a trauma surgeon in China and after graduate from military medical school. And I work with one of the founders of maybe trauma surgery in China, Professor Wang. And that time is mainly on what they call the blast injury. It's like explosives and severe damage, and to see how that causes damage and how the tissue repair. And then after that, I went to London to work with Jeff Bernstock and molecular biology and biochemistry at the University College London. I still think I would go back to China to continue my clinical work. So I kind of tried to look around for a job, look for a more project which is more kind of close to India. Sorry. So I found Professor Colin McCaig and John Forrest at the University of Aberdeen, and I went there. And that time, Colin is working on like tissue regeneration of the central nervous system. So he's using electric fields to guide neuron growth, like neurite growth. And John Forrest was working on corneal wound healing. So it's a kind of perfect combination for me looking back. And I combined the electrical signaling together with the wound healing research, and it works well. So I think I should avoid getting other people into the. So that's why bring me to this electrical regulated wound healing. And at the beginning, we were only trying to understand whether this is one of the factors in healing of the wound. But eventually, we found out that the electric signal can override other guidance mechanisms. And that kind of prompted me to think more. Not this is only one mechanism, but could be a predominant mechanism, maybe a conserved and ancient one. If you look at the wound healing, you think about the single cell molecules, plant or animals from salamanders and others, lower organisms to us as a human. And we all heal after wound. And what is the conserved key thing is that the breakdown of the barrier of the inside and outside. And the immediate effect is the communication between the inside and the outside. And we know that the inside and the outside of the animal have this chemical gradient as well as an electrical gradient. And so this electrical breaking down of the barrier is the same mechanism for wound in plant, in single cell, and for all the animal species, and to us as a human being. So this electrical communication might be the initial signal that telling the organism that there's a breach of the barrier and we need to repair it. And when the barrier recovers and this communication stops, this wound electric current would stop. So that time I was thinking I probably need to move to a bigger place to understand the fundamental biology of it, and then to develop this, to use this mechanism to help wound healing. So that's kind of prompted me to move to University of California, Davis. And even before that, and Mike and I knew each other's work, and we think this really is a fundamental mechanism, not only for wound healing, but as Mike showed, for many for developmental biology, for cancer, and for other many other biology too. So this is probably, I don't want to go too long, but this is a kind of a short journey of why I get here.
That's fascinating. Well, thank you for sharing that. So we want to dive a bit into the history of the field, and well, we certainly want to understand the history of the 1900s and how the field has developed. I know you've written a bit about some of the origins of bioelectricity as well, going back to Galvani's days. Could you touch a bit upon the origins of the field?
Um, I think Mike probably is much better person to talking about the origin of bioelectricity. Uh, so Mike, maybe.
Yeah, I guess maybe to put it another way, as you were developing your work in wound healing, because I think you actually your work in wound healing is quite unique. It's very different from what we do, and it's very different from what other developmental biologists who do bioelectricity in the field do. And you can talk about what are the inspirations, as you think about the bigger questions of like how is wound healing, how is it controlled? What are the evolutionary, do the different the types of wound healing that you find, for example, in amphibia, in mammals, and so on? Are they the same? Are they different? What kind of background in that field informs your thought? How do you think about these things?
Yeah, so if we're talking about general wound healing in human skin or mammalian skin, and this actually went back almost 200 years ago about this du Raymond, a German physiologist. He was the founder of the modern electrophysiology. He actually measured action potential, and that time is not a single axon action potential, but it's a bundle of axons together. And if you stimulate one end, the action potential would propagate to the other end. And this is so he is the first one who discovered that. But almost at the same time, he trying to see whether there's any current flowing in and out of the skin breach or wound. And he found that. And so it is not a new discovery. If we go back to Galvani's time, and indeed those kind of the caught nerve and a piece of frog skin, and at the wound edge, there would be this wound electric current. And Galvani used this as a source, but that time the understanding is very limited. So he thinks that this like animal electricity is a kind of imaginary vitalism. But now we understand it's like flux of ions, and actually the tissue cells pumping ions and generate and maintain this electric current. So if we go back, and you can see that although the understanding is not really as correct as we see it now, but the phenomena or the discovery, the observation were there. They found out that whenever you breach an organism, and there will be a kind of flux of ions, and eventually we see this as a kind of flux of specific ions. This is a kind of different from the electricity we use every day in our electronics and in our appliance. That the electricity generated by the power plant actually drive the electron flows through the conductive metals. But in in biology, it's more flux of ions, different, like sodium, potassium. So while the understanding getting more and more detailed, scientific, but the observation of the electricity in wound actually went back to hundreds of years ago. And if we, as Mike just mentioned, if we look at the wound of single cell, if we have a single cell, if you punch a hole, do we have electricity there at the wound, the primitive wound? And they are. We measure them. And other people measure it too. So early days, I probably lost century, about nearly 100 years ago, people used this big axons. They make a cart, and they can measure this injury current flowing out and in. And we know now, because you have a membrane potential and you cut it, and you can measure this current flowing in and out, well, this flux of ion in and out, and this generates injury current. And with a small, what was like frog, with frog skin and many other tadpole skin, and if you make a wound, this flux of ions happened, and this would generate the electricity. And now to human beings, this electricity generates more or less in the same principle. But if you look at the so this is the question. I go back to early days where I start to work with Colin McCaig and John Forrest, and I'm always wondering whether is this true? And many other people questioned about electricity. What is it, and how it would happen at a wound? Is it really important? And I kept asking myself too, and this. But if you look at a single organism, an evolved multicellular organism, and to us, this barrier inside and outside always there. And if the initial bridge to the single cell membrane happened, and the cell either reseal it or the cell will die. And so this would be a cue or a signal that happens so ancient in biology. And I think biology, the evolution will take the cues from the nature and say, well, we'll develop a mechanism to use this signal to develop, to heal. And so the biology can evolve and the complex organisms originate. So although specifically with skin wound or the epithelial wound, we have this electrical signal happens. But if you go back to the evolution to the very start of the life, and this bridge of the membrane or the barrier happened at the same time, when this flow of ions happens, and that is intrinsically related to electricity generation at wound. And so I would think if the biology doesn't take that as a mechanism, it doesn't go well with Darwin's evolution. And we know the evolution is just biology will take any cues to get it better, to survive, to repair, and to evolve. So that made me believe this is fundamental mechanism. Although we focus on wound healing, but I think for many other aspects of life, it would be hard to imagine that the life doesn't really, well, ignore this intrinsic aspect.
So I guess if we go back to when you entered the field, and this might be more of a general question, but how would you describe the state of the field then in terms of the major thinking in the field or the experiments that were being worked on, or just even the excitement around bioelectricity?
I guess probably this field is like many other fields that sometimes it seems it developed quite fast and well. And sometimes you need to overcome many hurdles, like those ideas already well established in the field, and to get people to look into this direction. And sometimes I'm quite optimistic, and sometimes I'm a little bit skeptical that it might take time for people to appreciate this mechanism. So specifically to the wound healing field, and I think people tried use electrical stimulation to enhance wound healing long time ago, and with many different approach. The misconcept is that you stick electrode there and you would stimulate the wound to heal. But the biology itself is far more complicated than that. So I think one of the the argument supporting you use the electricity in wound healing or regeneration is that it's cheap, and a few $100s, and you can stimulate the wound to heal. I think we need to think carefully about that statement. Nowadays, we know that if you develop a drug, you need millions of dollars to develop, to understand what is really happening behind, well, in biology system about this chemical. So if we just want to spend a few, much less amount of money and develop an effective therapy, that is not right. And also because electrical stimulation is so easy to implement in clinical context. Well, although not correctly in most of the case, but people publish different result, and some say, well, this is a magic treatment, and the wound never healed or stay there for many years, and after stimulation heals. And some people say, well, this doesn't really have any effect. So if you think about the wound complexity, and this is quite, I think this is quite quite understandable. The wound is geologically complex, chemically complex, and physiologically complex. And this is kind of three-dimensional volume conduct. So when we understand the electricity, we always have metal wires and the electricity flowing from one side in direct direction to the other side. But if you thinking about the 3D conductive medium, the electricity go every direction. So, and this is much less well understood, and it's very difficult to study. So I think the promise is there, but at the same time, the community and the society need to understand that this is a new type of therapy, and the funding agency need to spend time and money and effort, rather than just think that, oh, this is a magic thing, so you stick the electrode there and the wound heals. Magically, you only spend a few $100 and the magic stuff will happen. But we start to gradually see people, wow, the funding bodies are willing to, well, it becomes more willing to invest more. But I would think there's still not maybe enough.
So I think part of what you're saying is that initially when people started understanding that electrical currents could heal wounds, they thought there's some magic formula here where if you just apply electricity, it's going to heal. But a lot of your work is involved in understanding how these electrical signals heal the wounds, and it's a lot more complex than simply applying electricity.
Yeah. And as Mike's work already shows that this electricity carried by ions, the charge, because the charge carrier in biology is ions. And the ions, we have so many different type of ions, like sodium, potassium, calcium, and so chloride, all these things, and they can change, and they are the biology system itself are trying to control it in many ways, use ion channels and pumps and tight junctions. So the equivalent would be the battery, the resistors and transistors. So this biological electronics need to be understood first before we really try to manipulate it. With the advance of electronics, and as Mike is leading the way, and bioelectronics, and I think we will get much better understanding of the bioelectricity itself. I think bioelectricity, the name, is being tarnished somehow by charlatanism, and some magic things happens, and the dead body and the move, and there are some science in it, but it's more than that. And it's like Michael Faraday many years ago showed this electricity and magnetism somehow connected by this swirling needle without touch. And you start thinking about that, how can you actually make those things work together as a system? And the biology found a way, and I think Mike leading the way to understand this, the biology behind it, and then not only for developmental biology, but also offers a kind of complete new understanding of other aspects of biology, including wound healing and regeneration.
Do you have any? I'm interested in I mean, talking about other prior work and all of that. What's your take on some of Becker's papers? So Robert Becker, he made some pretty striking claims about rat limb regeneration and things like that. What's your take on all of that? And he had quite a different model, actually, right, of how it worked. What do you think about that?
I think Becker's work is pioneering, and also I think it's quite important. It's kind of like somehow form the part of the foundation of our work now. But as I said, that some of the understanding may not be as simple or as accurate as we hope for or wish for. Although, so I think this need to go into direction, well, in parallel direction. First is to apply it as Berker's work, and the other thing is trying to understand it, and they go in parallel. I think at his time, the tools and the methodology are probably not yet to really make this in a way that we can understand. We can still try that, and many things in medicine is just try and error. So, for example, you try something and it works, and that's fine. Maybe if we understand it, that would be better. But if we don't understand it works, let's do it. But nowadays, because we develop such a powerful mechanism, way or technology to understand things, like Mike, we and other people are doing now more and more, and I think we can understand more. But I think for his early limb regeneration wound repair work, what lacks over there is mechanistic or well understanding, and also a well-controlled approach that is other people or other lab can easily reproduce and develop in a more scientific direction. And I think probably now it's a better time in that time. And we know when things develop at that particular time, probably we don't have those techniques or technology to understand or to get a better control.
On this point, who are some of the other scientists you drew inspiration from in the field or you built your work upon?
I think this can go well for the person I actually met, and I think Lionel Jaffe is certainly the one. I still remember when I was kind of the junior young postdoc in London and in Aberdeen. We met, and I always get so excited, but about this work, and he always say, "Calm down, and you need to really able to explain things clearly." And make other people excited, so you become a scientist. At the moment, you're just a technician, and you do a nice experiment, and you got nice result, but it's very difficult for other. So this is one of the example. He's a pioneer of the next level of the new era of bioelectricity, and he did really some pioneer work, and I keep going back to read his papers. So he's one, and later on I met Ken Robinson, Richard Nusatelli, and also Richard Borgens, and I think they all are contributing many important ways to this field, including developing the technology to measure the wound electric field or any type of electricity associated with biological specimens. And they measured the electric field at different type of wound, and they kind of lay the foundation of the future work. And also there are the other people to show that, or the other ones, Ming Pu, and he stopped working in this field anymore, but they are among the first few peoples demonstrate that they are electricity in biological system, and you can actually measure them, you can map them. And the second thing is that the cell do respond to such an electric field. And the other one is Claudia Sten, and this is probably more relevant to what Mike has been doing in biological systems, that they use this modern technology to map or measure this electricity around the developing embryos, and then see how that relates with the developmental process. So, and of course, Colin McCaig and John Forrest, and they're talking about this from the clinical observation of the patient eyes, this swirling movement of the epithelial layers in the cornea during healing process. And if this pattern is disrupted and the wound is not healing well, and how this special movement pattern happens, there's many kind of hypotheses, and one including is the electromagnetic guidance of the cell movement. So, and as Mike mentioned about the Becker's work, and I haven't met them, but I know the work. So this field's kind of course, Mike's work is leading the way, and I read his paper again, trying to understand. With my clinical background, I'm not knowing very well about genetics and molecular biology, so I learned a lot of the molecular chemistry understanding genetics from his work. So those work are the foundation and keeps inspiring me and my lab to push this forward.
I'm curious, do you know who were the people working in bioelectricity before Lionel Jaffe's era? As you said, Lionel Jaffe was sort of the new era in bioelectricity.
Yeah, and at the beginning, as I mentioned, that the bioelectricity, when it first emerged, it's like people see this magic effect, and a piece of tissue twitch, and a piece of muscle contract, and that animal start to move, and even the corpse move. So the sensation was there, but the real development didn't come as like Michael Faraday's discovery showing that this needle without touching it, it start to move, and Michael Faraday's work actually revolutionized, well, leading to the revolutionized the whole society. Now we see it everywhere. Without him, we would not be here. And then James Clerk Maxwell, he really connect this electricity with everyone, everything. And as Einstein said that James Clerk Maxwell is the giant. He stand in the shoulder and developed the relative theory. So those are also great people underlying our whole human society move forward. I kind of forgot what you asked for, sorry.
No, that's fine. I think that answers it. I was curious who some of the pioneering scientists were before Lionel Jaffe, but I think you answered it.
Okay, yeah, it was in a more broad sense, yeah. But in biology, there are many other like Mike mentioned, more clinical relevant people like Becker, and he's a clinician and tried many things. But it's kind of hard that when people looks at like more fundamental biochemistry, genetics, and so cell biology, and but now I think it's probably a good time that the bioelectricity gets more connected with what traditional understanding of biology itself.
I mean, do you want to go through a few examples? I mean, your work is really extremely impressive and very close. I think maybe you're the closest to clinical application in this field. And I'm just curious if you could go through a few, skin, cornea, all of the, like, you've done some amazing things. So like, what are some of your favorite stories so far that have come out of your lab?
Yeah, so this can go a little bit further in detail.
Yeah.
So as I said, when I left London to University of Aberdeen in very north of Scotland, and I was thinking that maybe after a few years and I finished my postdoc training, I would went back to be a trauma surgeon again, and but I remember the first thing is the cell movement in electric field is so dramatic. And that time when I talked to the people in the lab and I said, we don't have a video recorder, we don't have a computer to record it. And what I did is that I took a photo at different time point, like 0 hour, 1 hour, 2 hour, and I print them out on the photo paper and compare them to see whether they move. And the first experiment worked. And I see the cell move directionally. I was so excited. And that kind of made me stay there a little bit longer. And then we start to ask, well, at the wound, there are so many mechanisms we accept that stimulate and guide the cell into the wound to heal. Now you say electricity and how important this is. And there was a postdoc in my lab. He so we try to make a wound and apply electrical field across. And I remember that time we start to have the video recorder. We can see the wound edge move. For example, this is a wound, this is the one side of the wound, the other side of the wound. And what people normally do is to measure the area in the middle about the wound and to see how the wound becomes smaller. And after a few months, she never told me the result. And I keep asking her, and it turned out that she thought that I want to see the wound getting smaller. But what her result is that when she applied electric field across the whole field, and the one side moving, the other side move away. So the wound area didn't really become much smaller than the control, because the control wound actually moving in the heels. So she was quite hesitant to tell me the result. So I said, well, after this 2 or 3 months experiment, I would like to see the result. So she bring the video to me, and I start to see this one side moving under the guidance of the electric field, the other side being guided away. And I said, wow. So it looks like this is more important, because at this experimental wound, we have all other accepted guidance cues. But applying this electric field, you override them. So one side actually move away. And this is more important than I thought. So I said, well, this is not just make it go smaller, but it actually override the guidance mechanisms like injury stimulation, contact inhibition release, the population pressure grow into the wound, or the chemical release at the wound to attract the cell in. So this is not just one of the mechanisms. It seems that it overrides and being a predominant or master signal guiding the cell into the wound, mobilize and guide the cell into the wound. So that time I start to thinking about this. This is more, because at the wound you have so many different chemicals like growth factors. You could have hundreds of them. And if you add each of them there, you will see some sort of effect. But none of them are able to really make this piece of tissue actually go away or in as you change the guidance direction. And then we ask the question whether the electricity caused such effect is physiological. And we look into the literature, and we see the measurement from Ken Robinson's lab, and then Lucitelli's measurement, and Richard Bogan's measure, and the measurement, the field strengths are within that you can measure at the in vivo wound. So this physiological electric field are able to guide, to have this overriding guidance effect. So I start thinking, oh, maybe this is not just one of the mechanisms. It could be a predominant or master signal to mobilize and guide the cell, at least in the simple wound healing model. And we tested in kind of like a organ culture system, and this still shows the same effect. So that getting me more kind of excited. We talked about the fundamental mechanism, but this experiment made me thinking about this could be a very important mechanism. And then so this is kind of like a second discovery. And that time, nobody actually are able to use other guidance mechanisms like chemotaxis or durotaxis or any other guidance to cause a massive cell movement. So this is the other, I think, I remember well what come out from the lab. And then we start to thinking about, okay, there are many genetic and molecular mechanisms regulate the cell movement. Does this electric field or electric guidance actually use those mechanisms or intersect with those genetic and molecular mechanisms? So I start to get in touch with Peter Devotis at Johns Hopkins and Henry Bond at UCSF and Joseph Penninger at that time at the University of Toronto, because they all study this chemotactic effect and they use a molecular and genetic approach to see how cells migrate directionally in chemical gradient. So I trying to work with them to see. And funny thing is that at the beginning, when I contact them about this electric field guided cell migration, they are very skeptical. And we're thinking, wow, what electric field? And all the other ones, Vic Small in Vienna, and that time I was in Scotland, it's closer in distance. And they're trying to do similar experiments. It's very hard to repeat because they are, most of the lab members are trained in biochemistry and molecular biology. They are very, very good. They are leading experts, manipulate the chemicals. But with electronics is less, it's probably less familiar to them. So in the lab, it's difficult to repeat. But I said, well, I can come over to do the experiment in your lab. And I went there and we reproduced this electric guided cell movement. And then we start to use a different model organism like dictyostelium. We knock out the genes. And with neutrophil in Henry Bond's lab, we block out a certain signaling pathway. And with Joseph Penninger, he has this expertise to knock out the genes in mice, and we can use this cell and tissue to test and see how the cells actually sense or transduce this electric signaling into cell directional response. And that time we found out that the key molecules like the PI3 kinase or P10, those like important intracellular signaling mechanism, kind of participate in this electrical field guided cell migration and in wound healing too. So that bring our with their help and together with them, we start to understand more about this important mediators or molecular mechanisms underlying this electrical guided cell and tissue movement and wound healing. So those are, I think, the early experiment I remember in the lab. This is still bringing back a lot of very good memories to working in different places and working with different people, and people start being very skeptical, and later on becomes very, very enthusiastic. And like Peter Devotis, we're still working together trying to understand how this, for example, one of the project we'll work together is that the cells now are in an environment, they have a chemical environment, mechanical environment, with Wolfgang Lucet and University of Maryland, and also electrical environment, and how the cell integrate them and into a kind of cell behavior response. Although we are able to understand them in separation, the one by one, but the integration is a key. Well, I think we are not there yet, but I think probably with this integrated approach, we'll eventually are able to combine biochemical, biomechanical, and bioelectrical together to see how cells are collectively, well, make a collective decision and respond to such a diverse environmental cues. And we were talking about mechanical mechanism. And Donald Ingerberg is another person who inspired me. And I remember we spoke at the meeting, and he said, "Oh, keep going," because at the beginning when he working on mechanical things, and people were always wondering, well, the mechanical force out there, and the cells always get the chemicals, and they bound something, and signaling, and the cell respond. And when you're talking about mechanical, how do cell do it? And so he's a big source of inspiration for me as well.
And what are some of the questions that you are exploring currently?
Yeah, so when I moved the lab to the US from the UK, and I was thinking there's kind of three directions I would like to pursue. The first is that, I mean, we talk about the wound, we can measure the electric field. So how the wound actually produce such an electric field and regulate them? And we know that our tissue or the wound are composed by the cells, and the cell form a tissue, and the cell have this ion channels and pumps, and those ion channels pumps together with the tight junction, and they form a biological electrical circuit. And this circuit actually generates this electricity, and it come into being when the barrier is broken down, to change a long time, and then when the wound heals, it disappeared. So a fundamental question would be how the wound actually produce and regulate such a signal to best achieve wound healing. And so we're trying to see whether we can find what we call the molecular generator is like a little power plant in the tissue that actually generate this electricity and in response to injury. So this is one direction. And the other direction is that how the cell actually sense such an electric field. And this is a long-standing question, and many people in the field are working on that too. Because when I first see the cell movement, I talked with traditional electrophysiologists, and very disappointing to me is that most people say, "Oh no, they won't do anything because the field is so weak." Because if you need to induce action potential, you need a much larger field, much stronger field, and they don't do anything. But if you look at the video of the cell movement and the wound healing process, the cell certainly respond to it. And although, as I said, that working with Joseph Penninger, Henry Bond, Peter DeVotis, we discover some of the important molecule and genetic element. But we have not found a sensor yet. So, for example, we can see light. Light is electromagnetic waves, and our retina detect it, and we can start see them. So the cell sense it. But for such a small DC natured electric field, we don't know what the sensor they are. We have some clue, we have some indications, and we found out some of the mediators. But the sensor have largely been elusive. We have so this is another direction. We're trying to see whether we can eventually find the sensor. But maybe sometimes I'm thinking about Donald Ingerberg's saying about how the cell sense the mechanical force. And I remember one of his words is that the whole cell is a sensor. So maybe that's true. So this whole cell just sense it as a whole unit. It's not the traditional ways, like we have a molecule and it has a ligand and they sense it. So the sensor could be more complicated in just a single molecule. It could be a set of molecules. It could be the cell as a sensor. But it was still, as I said, working with Peter Devotis, and we're trying to manipulate genetically the molecule to see whether we can find the sensor. So this second direction. The third direction is is like working with Marco Rolandi from and other colleagues from Santa Cruz, and Rivka Izorov, a clinician scientist here at UC Davis, and together with Mike, we're trying to see whether we can develop some treatment to precisely regulate the wound electricity and deliver chemicals electronically to help wound to heal. So this is another direction, is that even if we don't understand fully the mechanism, how the cell sense the electric field, how the wound generates electric field, but we possibly can use it and develop technology to enhance the wound healing. Well, at the same time, we're trying to gain more fundamental understanding. So those are the three directions my lab are pursuing now.
You know, one of my favorite papers of yours is the one with I think the first author is Sun, where you've got the entire cells are going one way in an electric field, and if you fragment them into pieces, all the pieces are going the other way.
Yeah.
That was incredible. That was incredible.
Yeah. So I remember this experiment was conceived many years ago, before we actually are able to do it. I remember I tried it with Henry Bohm. So this was back in Aberdeen, and when I saw paper about the fragment of cell. So fragment of cells means that the cell doesn't have nucleus, just part of the cytoplasm come down, and this are biological significant to platelets is such a kind of structure, and if we now exosome, those are membrane circled structure with a little bit cytoplasm inside. And what Mike mentioned about this is that when we read those neutrophil actually release part of this cytoplasm as a little particle or little fragment in combat infection. So I went to his lab and I'm trying to do that experiment, but it turned out it's quite difficult to get enough cell fragment from neutrophil. So we never been able to get a conclusive result. And later on, when we were in Aberdeen, and Julie Theoret lab has this, well, the other lab showed, the other group showed that the fish keratocyte, they are very big. So when you treat them with certain chemicals, the part of the cell body comes off, and this little fragment still are able to move around. So we were thinking, oh, so this is a good example, and together with Alex Morguna, that time he was at University of Aberdeen, University of California, Davis. Later on moved to NYU, and so we talked with Dr. Sun and said, well, let's culture the cells and to generate this fragment, put them in electric field. And the result is kind of well, we never expected. So the fragment come off, actually move in opposite direction from the mother cell. And so this so it looks like the sensor and the response and decision making are kind of different part of the whole process. So the cell can sense it, and the cell can respond it. to electric field, but the cell also can make a direction to see whether they want to go left or right, or to anode or cathode. So this makes me thinking more about that what Donald Ingves saying, that maybe the whole system is a sensor. It just doesn't use a single piece of molecule and I sense it, I responded. It's like it's coordinate itself and make the decision to go to the cathode or to the anode. So yeah, thank you, Mike, for bringing that really mind-boggling experiment up. Yeah, we could never ever thought the result like this. We would think the fragment come up will move together with the mother cell. But no, they didn't. They moved the opposite direction.
Yeah, for me, we spend a lot of time thinking about collective intelligence and how the preferences of the collective emerge from the preferences of the parts, how the scaling works. And here you showed us a great example where the parts have a certain preference, but the collective, once you put them together, the collective actually has a quite different preference for its action. So I think that's a powerful example. Yeah. Well, thank you so much. This is a real pleasure. I mean, Min, you and I have known each other for a long time, but I really I think your work is amazing. I think it's the closest probably to the clinic of any of us in the field right now. And it's just it's beautiful.
Well, thank you so much.
Yeah.
Well, thank you so much, man. Really appreciate you taking the time. This has been amazing.
Yeah.
If you enjoyed the podcast, you might also enjoy my newsletter, livelongerworld.com, where I share practical longevity tips and also upcoming releases of my podcast episodes. So if you wish to subscribe, click on the link below or head over to livelongerworld.com. Thank you for listening, and I will see you next time.